Good morning, students. I am Dr. Ashok Stiroya, Associate Professor, Department of Zoology, SPC Government College, Ajmer. In previous lectures, we had discussed about the structure of muscle, the different type of proteins found in that muscle, and their functions and their functions. So today we are going to discuss about different theories of muscle contraction. The number of theories have been proposed to explain the mechanism of muscle contraction. The main theories we will discuss here is Zent, George's views, Devio's theory, and Huxley's sliding mechanism. The first theory of muscle contraction is Zent-George's view. Zent-George said that excitation of muscle is due to the presence of calcium ion. Calcium ion which build ectomyosin by joining actin and myosin. Second one is ATPase enzyme, which is responsible for muscle contraction according to Zent-George's view. The ATPase enzyme, which is found in HMM heavy myromycin, break down the ATP and transfer energy for contraction of LMM or light myromycin. Means the two things are very important in Zent-George's view. The first one is calcium ion, which make or which build the ectomyosin, and second one is ATPase enzyme, which provide ATP to break down, or which provide ATP and ATP transfer as energy in contraction. The second theory of muscle contraction is Davie's theory. This theory was proposed by R.E. Davies in 1963. According to this theory, a fixed negative charge is present inside chain of myosin filament and near the base of side chain. Means the muscles have the myosin and actin filament, and in myosin filament there is a negative charge inside the chain on the head and near the base. The third one is the sliding filament mechanism. This is the most recent theory as well as most accepted theory in the muscle contraction. This theory was proposed by Hugh Huxley and F.A. Huxley in 1965. According to this theory, during the muscle contraction, some structural changes do occur in the muscles. And now we would discuss the theory in detail to explore the mechanism of muscle contraction. The necessary force is developed by cross bridges present on the myosin filament. Cross bridges are produced or developed by the myosin actin bonding. The movement in myosin and actin myofilament make them slide on each other. In this process, the help of calcium ion and ATP also taken by the muscles, and the complete mechanism can be divided in 5 steps. The first step is initiation of contraction. Second step is activation by calcium ion. Third step is power stroke. Fourth step is role of ATP, chemical energy, chemical events in myosin head, and fifth and last one is relaxation. Uh, in this lecture, we will discuss only two steps. First, initiation of contraction and activation by calcium ion. How the muscle contraction initiated. It is also known as excitation contraction coupling because excitation and contraction occurs in a same period in a synchronous way, so it is known as excitation contraction coupling. When impulse reaches to neuromuscular junction by nerve cells, synaptic vesicles of synaptic nerve releases acetylcholine in the synaptic cleft. As we discussed earlier in neuromuscular junction lecture, there is a junction of nerve and muscle known as neuromuscular junction, where the impulse come from nerves to the muscles, where acetylcholine is secreted in synaptic cleft. Acetylcholine binds with the receptors lies on motor end plant and stimulate the sarcolemma of muscle fiber. It is known as depolarization of sarcolemma. This depolarization is also known as EPP, end plate potential. It reaches on entire sarcolemma. This depolarization reaches to T tubules, and through T tubules it reaches to sarcoplasmic reticulum. In muscles, the endoplasmic reticulum is known as sarcoplasmic reticulum. The sarcoplasmic reticulum, thus, after depolarization, approaches to sarcoplasmic reticulum. It releases calcium plus plus, yeah, or calcium ion. Uh, Means when depolarization reaches to sarcoplasmic reticulum, it releases calcium ions. Second step, activation by calcium ion. During relaxation period, active binding site present on actin where myosin binds are covered by tropomyosin. Means the tropomyosin is the structural protein which found on the actin where myosin binds. Myosin only binds when tropomyosins uncover the sites on actin to bind the myosin. This thus the bonding of tropomyosin, myosin and actin protein remains inhibited. we can say in this flow diagram that calcium releases to sarcoplasmic calcium release by sarcoplasmic reticulum, and it joined with troponin C. That there are three type of troponin: troponin C, troponin I, and troponin T. Troponin C binds with calcium ion, and it complex make a complex. This complex make the conformational changes in troponin I. Conformational changes when occurs in the troponin I, the troponin I is now troponin I pull the troponin T, which is bounded with troponin I with one end and 18 with other end. Means the troponin T is binded in both the ends, one end with troponin I and one end with troponin 18. 18 when it is binding with 18, where the troponin T is pulled, which is bound with the troponin I with one end and 18 with other end. By the pulling, active binding sites of 18 it happens to be open, and the actin and myosin bonding happens at that time. So the next three steps of muscle contraction, power stroke, role of ATP, and relaxation period we will discuss in next lecture.